Hi, I'm Hannah, and I'm an organic chemistry student at Wheaton College. Today I'll be sharing with you an article that discovered a new binding mode of a CGRP receptor antagonist. First, here's a summary of the article's main findings. This article, which was published in 2022 in the ACS Medicinal Chemistry Letters, presents the findings of Southall and Southall's team, who discovered that the diastereomer of one calcitonin gene-related peptide, or CGRP antagonist drug, is surprisingly just as potent as the original drug due to an unexpected way in which it binds to the CGRP protein. First, I'd like to give you some background. Why did I choose this article? I've suffered from migraines for several years. I've experienced how both the intense pain from the migraines and the secondary symptoms, like a stomach ache or strange vision problems, can prevent me from studying, from focusing on lectures, and even at times from being in class. This article caught my eye because it discusses CGRP antagonists, which are an available treatment for migraines. I've tried a few different kinds of medicines before, but they haven't worked well for me, either being ineffective or having bad side effects. So the research on more effective medications caught my eye. Additionally, I was inspired by the hard work of the researchers, their problem-solving skills, and the way in which chemistry can improve the quality of life for many people. The main problem here is shown on this slide. The calcitonin gene-related peptide, or CGRP, is a peptide in your body that has the function of controlling your blood pressure. If a person has migraines, however, their bodies create more CGRP than usual. This extra CGRP causes issues in your body by interacting with the nerves in the brain. The interactions between CGRP and nerves can greatly increase the intensity and duration of migraine pain. One of the possible solutions to this is a CGRP antagonist. An antagonist is a molecule that binds to a protein and limits its function. The three molecules on this slide are CGRP antagonist drugs that are currently on the market. Unfortunately, the development of these original drugs was extremely difficult, so something more efficient was necessary. The researchers in my article filled in this gap by thinking of a drug that has better binding stability, since binding stability increases the effectiveness of the drug. Their proposed solution was a macrocyclic structural design to provide the desired improvements to the original drug and hopefully have no bad side effects. Due to the limit of time, I won't be diving too deeply into the synthesis in this video, but this image shows a brief glimpse into how the steps are that the molecules were finally synthesized. In the top left corner, we have the starting material. Slowly, groups are added to it until it finally becomes the ending drug, with this big ring over here that makes it qualify as a macrocycle. Now that I've introduced this article, I'll be sharing its research findings. This image from Southall et al. Shows the macrocyclic CGRP antagonists along with their binding energies, or PKBs. That's the number that's right here under the structure. A larger PKB indicates a greater effectiveness as an antagonist, which means it's a more effective drug. The original hypothesis of the researchers was that macrocyclic antagonist number 4 would not be as effective as macrocyclic antagonist number 3 due to its difference in stereochemistry. As you can see, in the area of the molecule responsible for binding to the protein, the part on 3 that is a wedge is a dash in 4, meaning the stereochemistry is reversed. Further reasoning for the researcher's original hypothesis is that 4 has the same stereochemistry as 2, and 2 over here, as you can see, has a much lower pKb than the other drugs, indicating that it is not an effective antagonist. Therefore, the researchers thought that 4 would not be very effective. Surprisingly, drug 4 had a pKb of 9.1, while drug 3 had a pKb of 8.9. This was contrary to the original hypothesis. This surprising finding drove Southall et al. To explore and experiment more. They created a new potential explanation for the reason why drug 4 is so surprisingly effective. Their explanation was that drug 4 has a different way of binding to the CGRP protein than drug 3, making it just as effective even though it cannot bind in the same way. Here is a quote that shows Southall et al.'s careful consideration of this problem. The quote says, "This surprising finding prompted a thorough analytical reinterrogation of the final compounds, all of which confirmed that they were indeed different diastereomers, and that they observed CGRP pharmacology could be unambiguously ascribed to the assigned structures of the two compounds." This quote is important because Southall et al. Show here that they made sure to double check that the reason that 4 is different is of the same effectiveness as 3 is not just because they're the same molecule, but actually for some other reason, and that they are indeed different molecules, even though they have the same effectiveness. So here we are back to this original image that I showed earlier of the macrocyclic drugs. If you notice here, in addition to 1, 2, 3, and 4, I also have drugs 9 and 10 on this slide. Southall et al. Came up with a new hypothesis that if 4's effectiveness is due to its non-classical binding mode, then adding a methyl group to one of the nitrogens of both 3 and 4 to interfere with the classical binding mode, which creates molecules 9 and 10, as you can see, there's an extra methyl group on this molecule, would make 4 effective, but make 3 less effective. So essentially what they're trying to do here is add a group to the molecule that interferes with the classical binding mode. This would show that the non-classical binding mode is in fact being used because an effective drug would have to have another way of binding than the classical binding mode because the classical binding mode is being interfered with by the methyl group. The pKb's to the left over here and to the right over here show that this hypothesis was indeed correct. Ten was in fact even more effective than 9. Using X-ray crystallography, the researchers discovered that the binding site of CGRP bent itself to still be able to connect to all necessary functional groups of the CGRP antagonist, allowing it to bind despite the differing stereochemistry. Now I'll be sharing the conclusions from this article, my analysis of it, and a few final reflections. Southall et al.'s main conclusion was that because of its non-classical binding mode with CGRP, molecule 4 was effective as a CGRP antagonist, even though it had a different stereochemistry. This image here shows that the difference in stereochemistry ultimately affects the direction in which the amide points. This affects binding, interfering with the classical mode but opening the door for the non-classical mode. I learned from studying this article that although I have learned a lot of things through the semester, there's always more to learn. Through reading this article, I learned that developing even one drug can take many, many years. This has made me appreciate much more the medicines that currently exist. Moreover, I even began to enjoy this article as I got deeper into it and gained more understanding. There are a few future implications for the research discussed in this article. First, it opens the door for future CGRP antagonist development because now we know that there are multiple ways in which an antagonist can bind to CGRP. This means that there are multiple ways in which the function of CGRP can be stopped. Perhaps this new knowledge can lead to the development of migraine treatments that are safer and more effective than what currently exists. However, one warning is that before any drugs can be commercially made and used, future studies need to be done to detect how drug metabolism works for these new antagonists and their side effects. Thank you for watching, and please like and subscribe below.